Hello, I'm live, and today I'm practicing my presentation on DNA resonance and telepathy. I'm in Albuquerque in a hotel, and tomorrow I'm presenting at a conference which is called Tesla Tech Extraordinary Technology. Basically, lots of inventors with different devices, and some of them are therapeutic devices. So a great audience, first time ever I'm presenting this talk to such an audience. So now I will share my. So join me. The link is located at. The link is located at Hucolo main group at the Facebook. Now, how do I share my screen? Entire screen share. Okay. Now I'm doing F5. Let me see if I can see myself. Just a second. I should have done it before, but it's pretty easy to check. Hucolo main group. Opening. Okay. Watching. I'll just watch myself. Watching is not working. Join. Not working. Oh, that's sophisticated. Anyway, I assume you see me. All right, I will start my presentation. I have my timer. Oh, I know how can I see. Okay. Yep, everything is good. So I'll continue my presentation. So I will talk about DNA resonance therapy and telepathy. I'm Max Aug, and I'm from San Diego, and here are my contacts. Prepare your questions. My talk is made of 3 parts: DNA resonance code, and telepathy. And I will take questions in the breaks between 3 parts. So let's start with experiments on non-chemical signaling. There are fish embryos in a quartz cuvette, and you have 2 quartz cuvettes with fertilized fish embryos. Some are very young, they just fertilized a few minutes ago, and some others were fertilized 5 days ago, so they are older, they already developed, somewhat developed. If you put one quartz cuvette on another and let them grow for a day, you will see that the older embryos repress the younger ones. And of course, as a control, you use the same 2 cuvettes and you put a list of aluminum foil, and you will see that aluminum foil blocks the transmission. So these experiments are done by Burlakov, and this is one of the most reproducible, most published experiments which are available around. Here is the literature on remote cell-cell communication. There are experimental papers. I just picked a few papers on fish embryos, cell culture, and plant roots, and also there are reviews which I recommend. The pioneer of the cell-to-cell communication is Alexander Gurevich, and he was nominated for Nobel Prize 11 times, never actually got one. And if he would get it, that would change the history, could change the history of the humanity and history of science. But basically, the materialistic chemical concept took over, and field concept was buried. So he introduced a term, morphogenic field. Morph is a form, and genic means creating. So morphogenic field is a field which creates a form. So the idea is that when we develop from one cell into an organism, there is embryo development. This embryo creates a field which controls the shape of the embryo, tells it what to do. So that field is called morphogenic field. Also, he introduced the term 𝗪𝗘𝗕𝗓 radiation. 𝗪𝗘𝗕𝗓 radiation, and this is the radiation which is radiated by fast-growing embryos or tissues, and it induces other tissues to grow faster. So 𝗪𝗘 is from mitosis, so radiation which induces mitosis. Here is the experiment by Gurvich, one of the classical ones. It was reproduced by others. So you take the onion. Onion is good because it's a clean system. It has nutrients in it, and you can see the roots growing very fast. And you capture the light which comes from the root and direct it with a prism to another root of another onion, and you measure with which speed it grows. And using the prism, you can calibrate which. So you use prism to split light, and you can calibrate which wavelength is directed to the receiver root. And he discovered that those wavelengths lie in the area of around 200 nanometers, which is UV light, ultra weak UV light. So the experiments of Gurvich started around 1912. The main discoveries were done 1922, about 96 years ago. And they continued pretty successfully for some time until 1948, when there was a lot of repression in science and in Russian science as well. And the lab was closed, but it was reopened later in 1962 by his daughter, and experiments were reproduced. And now Burlakov's lab is continuing these experiments for the last 30 years and publishing over 100 of papers on those effects. So typical principle is when you transmit the signal from one container to another, and then you can put a filter to separate and measure and test which wavelengths are responsible for the cell-to-cell communication. And also a very interesting experiment when I can put a mirror and the sample is reflecting the light, and the mirror is reflecting the light from the sample, and the sample is affected by its own irradiation. So the experiments show that the radiation is basically in magnetic spectrum, and a germanium mirror interestingly accelerates the growth of fish embryos. So if they look at the mirror, the growth is accelerated, which is interesting. It means that the mirror modifies the rays in some way. And also there is another fancy mirror which is called retroreflector, and this retroreflector actually represses the development of the fish embryos, and causes developmental abnormalities. And it is very important. Basically, this proves that the effect is morphogenic. So we are dealing with the morphogenic mirror. So I ordered those quartz retroreflectors and already obtained the germanium mirror, and I'm getting now the system with fertilization of fish embryos through collaboration, and we are starting those experiments soon to reproduce and advance them. So my master's is in DNA chemistry 86, PhD in molecular biology 94. I did a postdoc in America for 10 years in genomics, gene regulation, animal models. And since 2008, I am working or running biomedical startups, and I'm funded for therapeutic electromagnetic devices targeting DNA. DNA is tangible. You can touch it. You can see it on a gel. You can sequence it, so it's very digital. These individual bands are 4 letters of the DNA. Now there's a multiple samples sequenced. So each band is a monomer, a nucleotide of the DNA. And you can crystallize and measure its prove its double helical properties using X-ray spectroscopy. DNA is very pretty. It's a double helix, and the key structures are inside the double helix. It's available. The sequence of the human genome is available since around 2000, already for 17, 18 years. And the best, friendliest of the genome browsers is UCSC genome browser in University of California, Santa Cruz genome browser. And you can access it freely. It's a public, it's in public domain. And you can access human genome and compare it to rhesus, mouse, dog, elephant, chicken, and so on. There's lots of genomes and lots of annotations online. It's all available for public use. An interesting part of it is, so this is a gene. The blue side part is a gene. And the gene is basically what is transcribed into RNA. So DNA is transcribed into RNA. Then RNA is translated into protein. So those vertical bars are little pieces which are translated into protein. So this part of the sequence of the genome we understand really well. We understand the code, how the nucleotides code for the amino acids in the protein. And it is about 1.5% of the genome. And the rest is still a mystery. We don't understand what it does. So there is a part which codes for RNA, but we don't understand the language of this RNA. We don't understand the function of it. And there is a lot of other part where there is no RNA coded. So this is called a non-coding DNA. So understanding that, what it really means, is still a challenge. And we'll talk about this challenge. Most important slide, take-home slide of this, you can access actual sequence, and the sequence is very simple. It consists of 4 letters, A, T, C, and G, and you can just read it, A, T, 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 A, C, C, and we don't understand what it says. There is no translation yet for non-coding DNA. Genome problems. The problems with understanding the genome. So basically, this is the development of the fish embryo, and you see it starts from a single cell, divides to 2, to 4, to 8, 16, and then there is a lot, 128, for example. And then there is a miracle happening from a blob. It converts to a shape. And the shape develops pretty fast, in hours, and makes something which looks like a fish. And it's similar to all life on Earth. How it happens, part of it is understood that genes are important, and part of it is understood that chemical signals are important. But we believe that chemical signaling is insufficient to explain biological development. And there is not enough genes in the genome because we have only 21,000 genes. It's approximately what other life forms have on Earth. So more complex forms don't necessarily mean more genes. So that's the problem. The genome is not understood. Now, the first well-described idea, which is known to me, on holographic genome is 1973 by my friend Richard Alan Miller. And he pronounced there all the ideas that the genome is making the morphogenic field. That the record is holographic. Basically, a linear DNA from each cell creates a third-dimensional holographic projection of the body, and this projection drives the chemical reactions, drives the development of the embryo, supports the health of the organisms and homeostasis of the organism. He did it 46 years ago with co-authors Burt Webb and Darton Dixon. This book was the one which I was given by a friend in 1999. So it's over 25 years later, I got this book by Peter Garyaev, Wave Genome. So it outlined similar ideas. And I spoke, and Garyaev is helping us a little bit. And the next year, there was a book by Marco Bischoff, and he pronounced all the ideas that DNA makes a dynamic web of light. It controls morphogenesis, and it creates a field which is a holographic biphoton field of the brain. So the brain and DNA are linked into one field. Very important. Konstantin Mail recently, 9 years ago, published a book, DNA and Cell Resonance. And here he proposed some of the physics for this morphogenic field. And this physics is basically, it is electromagnetic field, but of unusual nature, not classical, non-linear nature. Jacques Benveniste and Luc Montagnier contributed to understanding to proving that DNA can be converted into the field. I mean, the DNA can be induced to make a field which can be captured and recorded by a device, transmitted to the next device, and this field can be sent to the another sample, and the structure of DNA would be transmitted technologically from one sample to another. Now let me define the DNA resonance. So resonance is when identical structure resonance. The idea of the DNA resonance is that identical structures resonate, meaning they synchronize and they can transfer information. And the typical example of that, if you take two guitars and you start making sound on one guitar, then the second guitar starts resonating. It's quieter, but you can transmit information from one string to another using resonance. And for DNA, it would be two similar sequences, TAA, CCC, the sequence of our telomeres, would resonate in different cells. So that's the definition. So the idea is that identical identical structures resonate. Identical sequences should resonate. And here, expand a little bit the idea of DNA resonance. It could be a resonance oscillation of different structures capable of similar oscillations. So here, for example, a guitar and a sax, and if you make a note on a sax which is present on the guitar, you can still transfer information and the guitar would resonate. So it's not necessary that they have to be identical, as long as they can resonate, as long as they make similar sounds, identical sounds. So here is an example in DNA, like TAA, CCC would resonate with, as I predict from modeling, with CAA, CCC, if C is methylated. So these structures would be having similar electronic oscillations, although chemically they would be somewhat different. All right? So this is a slide on DNA resonance, which I presented at a talk in Boston 15 years ago. So the idea is that chromatin structures may transmit and receive signals. And here I just mentioned the novelty here is that we remember that in our cells, the DNA is not naked, it's wrapped around nucleosomes. It's packed. And I suggested that nucleosome would be the unit which transmits which resonates and transmits the waves. And as we do it in our cell phones, you need to use energies to send the waves, and we need to use energy to receive the signal. And in our case, it would be a protein binding to the signal, would be a chemical signal transformed into the electromagnetic signal, and then transformed again into chemical signal. And the energy usually we get from ATP. And now there is some evidence that there is a lot of ATP processing enzymes bound to DNA. So it's pretty much likely that it is the case. So this is a summary which I will repeat along the talk. It's pretty well summarizes pretty well the idea of the talk. So the DNA is part of the biochemistry. It produces morphic field and interacts with it back and forth. The morphic field builds the body structure and supports it, sustains it. It guides the homeostasis of the and morphostasis of the body. It also guides the biochemistry. Morphic field guides the biochemistry. It also guides the behavioral biochemistry. And it is guiding the mind and performing some of the mind functions. So mind is electrochemical plus the field, both. All right. Now, if you look at the ancient teachings, you can put a correspondence between this part of the morphic field, which is responsible for health, is called etheric body sometimes, and which is responsible for emotions, is called emotional body, and responsible for mind is called mental body. So these are different frequencies or different oscillations of the same field. So there is a pretty good agreement in this idea. Now, the next idea is that our mind morphic field could be personal and also could be collective. Sometimes we get communications that go through the collective field, and the experiments that proved it with statistical significance, with exceptional statistical significance, are done by Sheldrake, and it's also a long time ago, 30-something years, 37 years. Now, so the idea is that mind and body structure interact with individual morphic field and collective morphic field. So here I try to put some measurements, some basic physical measurements on the fields. So the body structure, biochemical factory, behavioral chemistry, and mind are electrochemical. Mind is electrochemical. Behavior is electrochemical. Body is electrochemical. Now the DNA, let's keep it for now. Now the morphic field is this big green box. It consists of 3 dimensions or 3 types of the field. One is electromagnetic, the one which is familiar to us. Another one is fine electromagnetic. I call it quantum physical, but there is many ways to describe it. Some people call it scalar field, and you know all the names which are involved. So these are the field which is borderline between our dimension and the next dimension. And another dimension is something which is not accessible to our measurement tools yet. But the collective morphic field is there. And the reason we know about it is through after-death experience, telepathic experience, extrasensory perception, ESP, and so on. So there is a lot of happening there. And what is interesting, very often it is collective and it is not bound by space. So some of the signaling is transmitted from one person to another, like say in ESP signals, where the people are completely insulated, isolated from the electromagnetic fields. Still the signal passes through and there is no, it's not restricted by the distance. All right. So, and the DNA is crossing all of those fields. It is certainly biochemical. It is almost certainly electromagnetic. It is actually conducting electricity and conducting, and I claim that it is magnetic as well. It has magnetic oscillations. And obviously it is of the size which is between macro and quantum sizes. So Nucleotides are pretty small and they're measured in nanometers, in angstroms, fractions of a nanometer. And that's where quantum chemistry is happening. So I would say morphic field happens between electromagnetics, quantum, and beyond. All right, I will take a question in a minute. My research, I was excited about red light therapy because small amounts of red light produce a lot of effect. And I was measuring the DNA expression, conversion of DNA into RNA. And some of the genes indeed were activated by red light. And also was interesting, the effects of red light were very obvious. I could tell which mice were irradiated and which were not. And also we tested the effects of red light on skin cancer. And I hope that red light would cure skin cancer, and it didn't happen. Actually, there was a balanced effect because we irradiated the whole mouse, the tumors were activated as well as the rest of the body. So I would say this. We concluded that there was no statistical effect on the tumor growth, but obviously it was absolutely obvious that the mice which were treated with red light, although they were sick, they behaved much healthier. They were happier and healthier, while the other ones were nearly dying and very sick, behave like sick mice. So now I am moving to millimeter wave therapy. It's another frequency. It's 42 gigahertz, 7 millimeters. It's much longer wavelength. And it also has very interesting properties. And it's very popular in Russia and Eastern Europe. We estimate that over 1 million people have been treated with it, and it's available for purchase, inexpensive, and many people treat themselves at home. It works for pain relief, arthritis, and the effect is very systemic, although applied to the acupuncture points. In progress, I'm testing where this 42 gigahertz induce DNA compaction in cells, and the preliminary data is that indeed it does. So millimeter waves, 42 gigahertz, affects the DNA, and we're using some fancy Russian generator between 37 and 50 gigahertz. This is the spectrum, and I just wanted to highlight their UV range, which was highlighted by Gurvich as biologically active. LLT range, red light therapy, millimeter wave therapy, ultra high frequency therapy, also very popular in Russia, and sound frequency therapy. And you can see that radio, radars, microwave cell phones are somewhere in between, interspersed. Any questions so far? Let me see if there is anybody around. No questions, I continue. Part 2, how to crack the DNA resonance code. It is accepted by everybody that the DNA works as a computer. It's a very good analogy. It has a program. It is a program. It has memory. It is memory, and also there is read-write capacity on the DNA. So on top of the sequence, you can methylate it, and the body does that. So that each cell has different pattern of modifications. We hypothesize that also there are waves which are involved in compacting, decompacting DNA, and this is also a way to read or write on DNA using wave patterns. Also, it has logic, it has chemical circuits and conditions, and we hypothesize that it also has electromagnetic circuits and conditions. It has perfect ability to address and search for certain sequences. It's widely used in biochemistry. And another last moment is that the code of the DNA could be fractal. I'll pause for seconds if I can. Let's see. And I continue. So the idea of the fractality is that smaller pieces are proportionally equal to bigger pieces. So there is a proportional copying, and the smaller pieces are have the same proportions. And the big proponent of the fractal principle in DNA is Dan Winter, and I'm thankful for his help in understanding this. And what was interesting that Eric Lander, from one of the leaders, mainstream leaders of the human genome sequencing, also recently published a paper on the fractal principle in DNA. It's not in the code, but more in the packaging of DNA. But still, the idea of fractality is present even in the mainstream science. And what is exciting about Eric Lander, he often publishes the papers which become mainstream only about 15, 20 years later. I observed it continuously that when there is some trend in science, that Lander published about 15 years ago something on that topic. The holographic principle of DNA. So basically that's the idea of redundancy and damage tolerance. So here are the QR codes which were all are readable, but you can see that there is a lot of resistance to the damage. So they can be distorted in many ways. Some parts can be deleted, and you can still read them using the phone. So that's one part of the. So in the DNA, we also think that there is a lot of redundancy, and the code is spread all over the DNA. And in mainstream science, people also understand that the mind is also spread all over the brain, and I suggest that it's also spread all over the body. Another part of the DNA code idea is that it is holographic. And the idea of holography is basically the field interacts with the substance, and through that interaction, interference, they create a third-dimensional image. And in our case, it would be the image of the body and the image of the cell, the image of the enzymes, and this image would be dynamic, and it would move things around and control the processes. That's the idea of holographic genome. Now, how do we decipher what is actually resonating in DNA? So the candidate sequences, the candidate parts of the DNA are the double helix, and it's wrapped around nucleosomes, and there are groups of nucleosomes. So these are all candidate structures of different sizes. And even a chromosome is also possible, a big, relatively big resonator. So here we have about 20 bases, and here we have about 100 million bases. And still those could be like different resonators. So the frequencies of DNA resonating could vary a lot. And also the modes of oscillations could be also different. Could be twisting, bending, moving back and forth, and so on. Here is my one of like recently published models of oscillation of charges in the DNA. So DNA is a conductor, and here you can see that the charges could travel in DNA back and forth, creating an oscillator, and pink are the magnetic poles of the nucleosomes. So the polarity would change with each cycle, each half of the cycle. It's one of the possible models. So now let me pronounce the idea of the DNA resonance code. So basically, it is the idea of that algorithm that you take a sequence and you should be able to read it and produce what it says. So for the elephant sequence, which is 3 billion bases, you would produce an image of an elephant and all the description of the processes and behaviors of the elephant, biochemical process and behavior. So a lot of complex information packed together in one holographic pattern. So we already are familiar again with QR codes. There is an algorithm, and you can code the word elephant in a QR code, and the algorithm could transmit it back and forth. And the difference here and there is huge. That it's not one word, you can encode much bigger message. So how to crack the DNA resonance code? Is it crackable? I suggest it is, and the methods would be experimental genomics, when you modify the genome or you sequence the genome. It's very well developed modern technology, and it is widely used for drug discovery. It can be used for DNA resonance code discovery and understanding. Spectroscopy, when you take a synthetic piece of DNA and analyze its vibrational patterns in a spectroscope. Quantum chemistry modeling, basically you can look at the positions of the atoms, positions of the electrons and protons, and model the oscillations in the DNA. And computational genomics, there is a lot of interesting data already accumulated. Sequence data and functional data, which of the sequences, what they do, and in which conditions are they open, and when they're condensed, and when they're working, and when they are chemically working, and when they are not. And of course, linguistics. Maybe we can just look at the genome and discover the language just by trying to read it in many different ways. This is just to remind that there is a lot of public information accumulated in the databases. You can see which DNA is open and closed. You can see which sequences are bound to the nucleosomes and so on. So these are all these methods coming together. So the principle that similar molecular structures resonate can be used to combine quantum chemical modeling, annotations, basically the public data of the genome, functional data of the genome, and experimental approaches and spectroscopy. And as far as I know, no one is doing that yet. It is something which is up to grabbing. So we have examples of people doing that in the past. So Marshall Nirenberg, in 61, many years ago, 57 years ago, cracked the first letter of DNA code for the proteins. He took the poly-U sequence, added the proper enzymes, and it was converted to phenylalanine, polyphenylalanine peptide. And this way they discovered that 3 nucleotides UUU were coding for this phenylalanine. And after that, it took very little time, or maybe a couple of years, to crack the rest of the code. So once you crack the first letter, then it's downhill. And we have, for our resonance code, we already have some candidate letters and sequences. So we just need some experimental proof and computational proof that these are what they're actually doing. Now, Watson and Crick in 52 cracked the double helix. And what was interesting that both of them worked basically, neither one of them was funded to do that. And Watson was doing that for his own money because his supervisors refused to fund his research on this direction. And Crick did it at spare time while his bosses wanted him to work on something else, which was much more boring. So these are typical hackers which were able to create the greatest biological discovery of the century. And Einstein discovered his theory of conductivity, of relativity, when he wasn't funded to do that. He was also a hacker, and he hacked it just gracefully. Rosetta Stone was discovered by the Egyptian language was deciphered by Thomas Young, who deciphered the first letters, and Champollion, who was able actually to read the text. And the key for that was the Rosetta Stone, where they had 3 different languages, one of them forgotten Egyptian, and 2 others were known. So based on comparison of the text, they were able to start deciphering learning the language, deciphering the code. So we need a Rosetta Stone for the DNA resonance, and I believe part of it is in UCC genome database. I want to mention Craig Venter, who used another approach. So basically, because he wasn't funded by public and federal money, by academic money to do his research, he found some private money from pharmaceutical companies and some investors to compete with the human genome and basically finish it first in a more elegant manner. And his Celera also finished many other genomes, mouse genome and many other genomes, very fast. So their formula for the business was that they would patent the sequences and then make the sequences would be converted into drugs, and the drugs would pay off. It happened, but happened much later. But still, some of the sequences are now making the revolution in the pharmaceuticals, which are called biologics, which are very useful for arthritis and immune disorders. So how much effort is needed to crack the DNA resonance code? I would say if in 5 years, if my group gets funding for 5 dedicated postdocs, if we have a postdoc on computational genomics, experimental genomics, quantum chemistry, physics, and spectroscopy, we should be able to crack the first letters of the code. We are in a unique position because we know where the others got it wrong. In mainstream science and even in an alternative field, there is a lot of ways to interpret the data, and we have a unique grasp on what has been done using, basically we did a lot of homework. So the applications, therapeutic applications, so morphic field, body structure, and so on. For body structure, the applications are obesity, obviously. If you can manipulate morphic field using electromagnetic devices like electromagnetic drugs, you can cure obesity, cancer, and do a lot of organ engineering in a smart way, talking to the organs in their language of the morphic field. And then you can treat depression, schizophrenia, and Alzheimer's and Parkinson's because you would understand the language. So the devices would look like that. The only difference is that now they would talk to the body using the language of DNA. Questions? Part 3, the brain code. So part of that picture is that DNA is connected, is making a morphic field, and this morphic field is mind, is connected to the mind, it is mind. So DNA resonance code should be the same code as brain code. So these would be parts of the morphic field. This would be very closely connected. Notice here that unlike in many other fields of alternative science, here is this part of the science is heavily funded by the federal government, Brain Initiative, and even before the Brain Initiative, but recently it was boosted with Brain Initiative. So NIH, National Institutes of Health, DARPA, YARPA, and other agencies are funding the research on brain-computer interface. And what I noticed was that brain-computer interface could easily be expanded to brain-computer-brain interface, which can be defined as telepathy. See? Think about it. So the mainstream science understands that DNA sits in the nucleus, wrapped into the nuclear membrane. And here is a neuron, and there are sensory and motor neurons and some other neurons. and they're very long. They are several feet long, and the body of the neuron, this little thing, this body, could sit in their spine, and the endings would be in toes and fingers, in the skin, and the signal is transmitted through action potential, one way or another. And we believe that that's a simplistic understanding of the brain, and the rest of the neural system is insufficient because our mind is much more complex than the number of neurons can explain. And the speed of the action potential transmission is not sufficient to explain how the mind works. We need some other, more sophisticated actors here, and obviously one of the candidates is the DNA. And Stuart Hameroff came with another candidate, which is microtubules. And he suggested that microtubules transmit and store a lot of information. So here I proposed that the mechanism by which microtubules could transmit the signal is by forming so-called proton highways, which are known in some other parts of the biology. Basically, these are structured water, linear water structures, where it is structured in linear fashion, and where the hydrogens are united and distributed through the cloud. So there is a certain uncertainty about the position of those hydrogens. And they form a proton cloud, and this creates a proton highway, which is supported and structured by a microtubule. It's only a hypothesis, but it's a very pretty one. Something inside this microtubule is specially structured, so maybe it is water. And that this is used to transmit information. And that's a little bit supported. So the fact that microtubules are needed for the function of neurons is absolutely well proven. So now we just need the reason why they need it for the function of neuron. So proton highway and microtubules. Here is another idea which is pretty much established. The DNA, this is the DNA, and inside of it there is no water, no single molecule of water. It is these aromatic structures called bases, nuclear bases, or DNA nucleotide bases of the nucleotides. So they form a lipid structure, a fatty structure. But this structure also has distributed electrons unified across the linear structure, such as in metals, similar to the way that they're unified and distributed in metals. And also, I suspect that there is also proton cloud inside of the DNA, also linear. So possibly we have a proton-electron highway inside of the DNA. So here is I published, I noticed, I suggested that DNA could be magnetic, and two strands of the DNA could have alternated magnetic directions. So they will look at the different directions. And I noticed that some of the bases of the DNA are single rings, hexagonal rings, and other bases are, they call indoles, so they have double rings. And in double rings, possibly there are two different directions of the magnetic field, so one and another. So if you spin it around in a helix, it would look like that. You just see, like, if you take this structure and spin it, wrap it around, that would look like that. So I said, and this magnetic structure will depend on the DNA sequence. So here is I suggest that's how the DNA sequence is converted to the magnetic pattern. And I think that's a terrific clue to the discovery and deciphering of the DNA resonance code. So these structures possibly are the key to the resonance code. It is published half a year ago, self-published half a year ago. Now, adding the Hameroff's idea, I combined the DNA with microtubules. And I noticed that actually my collaborator noticed, my co-author noticed that microtubules actually wrap around the nucleus, and possibly there is electromagnetic talk between the DNA inside through the membrane with the microtubules outside. So DNA resonates, sends the field to microtubules, and microtubules as wires transmitted through the brain and through the body. So the field could be not just created and shined outside, it might also be directed through the highways to specific places. That makes it even more interesting and pretty. So how to crack the brain code? So based on the DNA code, okay, brain code, DNA code, they link. So we have a good tools for understanding the DNA code. So just using experimental genomics, say on mice and other simple organisms, we can start cracking the vibrational code and get to the brain code. Also, we have some candidate sequences, so we can see if those sequences are effective in the brain code, basically in electromagnetic oscillations. And there are several other ideas which I kind of summarized here, but these are the main ideas. So we can come to the idea of the brain code through the idea of the genetics and genomics. And genetics and genomics already has a lot of tools. So by combining these experimental tools and computational tools, we can get closer to the brain code. Now, implications of cracking the brain code. What will happen after? So physical therapy for psychiatry. The devices, electromagnetic devices, which would treat psychiatric disorders, depression, schizophrenia, and so on. Mind enhancement devices. Unfortunately, the mind control also becomes more possible. And technology-assisted telepathy. And I want to highlight telepathy aspect especially. So telepathy I would define as communication from mind to mind without the use of physical senses, which is basically the Wikipedia definition. So the telepathy will improve learning and communication. So the teacher can talk to the student, or the other way around. The teacher can talk to the student and deliver information directly without using the limited language. So it is like working on slow internet connection and super fast. So telepathy would be a super fast internet connection, and distortions would be smaller because it will be mind to mind, wider channel, hopefully. Now, telepathy will improve the teamwork. So volunteers would combine into teams, and the telepathic inform. Telepathy will enable telepathic communities. Currently, our civilization is in a stalemate. Basically, there is a lot of crises going on everywhere. So, and I say that most of the crises, it's like zooming in, happen because people are close-minded and selfish, irrationally selfish. So here, say the stoplight broke, and people didn't follow the rules, and they got stuck. And if you look carefully, if this car moves away, this car move away, this cars can move, this cars can move, this cars can move. So if they had telepathic communication, if they were able to talk and define the positions of everybody, understand what happens, and develop a plan, and convince this car to temporarily move away, everybody would win. So telepathy will improve cooperation and promote altruism. Also, it will prevent deception. It's much harder to deceive when your brain is connected directly with the other brain. So in sales, people believe that you cannot do any trade and sales without actually deceiving others. Now I put here the helmets. So now people are connected telepathically, and a lot of trade would have to change. So in the telepathic communities, in voluntary telepathic communities, people would have to do things honestly. That would change a lot of things. What do you think? And it will prevent dishonest leadership. So the leaders also won't be able to cheat. That will solve a lot of problems. And finally, telepathy will increase connectivity. And this will lead to a shift in global consciousness. So we'll have a dimensional shift. So pros of telepathy are huge. And cons of mind control are outweighed by the pros of telepathy. And what is most exciting is that the interests of the humanity at this moment are in line with the interests of the brain initiative and the ideas of brain-computer interface. So finally, for a change, there is a possibility for alternative scientists to be cooperating on very interesting project, which is actually good for the humanity and get actually funded. So the approaches are DNA code, brain code, experimental genomics, genome screen, mind-to-mind interface. So approaches to developing telepathy. Again, just understanding that the telepathy comes from the brain code, is from the DNA code, is unique. Nobody in the mainstream is even thinking about it. So just understanding that there is a DNA code there, already defined, gives some answers. Again, we can use experimental genomics and animal models to start experiments on that. And I'm talking about proposing grant proposals. We can propose a genome screen and find the genes which are helping the natural telepathy. We can use some of the DNA microtubule approaches for mind-to-mind interface. And also we can use some technology to synchronize subjects using biofeedback and enhance the abilities for natural telepathy, expand them and enhance them using biofeedback technology. And by the way, we developed a prototype and it is ready for testing. So we would be looking for an official IRB-approved study on the telepathy using some biofeedback type of device. So would your device enhance the telepathy? So think about collaborating or applying for the grants, or collaborating with us and applying for grants together, and studying the telepathy and developing telepathy using advanced technologies. I would like to acknowledge the help of Richard Alan Miller in microtubules and DNA, of Dan Winter in fractals, Peter Garayev in understanding his experiments, and Alex Burlakov in us setting and helping us to set up our own experiments. Collaborated with microtubules, therapy, physics, neurobiology, and Ivan Savelyev was very helpful in developing all of those topics in the last half a year. Nelly Zariano works with me for the last year and a half. Jane worked in the past doing some experimental genomics, and Nelly and Ivan do mostly of theoretical work. And Jane Golovina is starting as a computational genomics expert. Here are my contacts, and I would enjoy and be very interested in discussing further actions, and also meet everyone and talk over phone and email and video communications like Skype. So we could meet later after the conference, and I would be delighted to meet you guys. And I invite more questions, and thank you very much. Have a good day. Right, so this was my presentation, which I'm giving tomorrow in the morning. Send me some love, and I would appreciate your help. Have a good day. And if you want to contribute or talk to me, I'm accessible on Facebook Messenger, and email is good. Good day. Om.